Hello, everyone, and welcome to today's webinar titled Reshaping Cell Biology Experiments for Automation. This webinar is a part of the 7th annual event in the Cell Biology Virtual Event Series. I am Kaylee Walk of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Automata. For more information about Automata, go to www.automata.tech. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen from the lobby within the event. Now let's get started. I would like to now present today's speaker, Russell Green, Director of Product Application at Automata. For a complete biography on our speaker, please find it on the left of your screen. Russell, you may now begin your presentation.
Hello, thanks for joining. My name is Russell Green. I am the Director of Product Applications at Automata. I'm stepping in for my colleague, Jeff Dance, who unfortunately is demonstrating that the pandemic is still with us to some degree and has fallen unwell with COVID. So wasn't able to join us for the recording session for today. And so we decided to share some content that we actually recorded just a month ago, which is very relevant to this particular space. And it's around how we both can modify our system designs of automation and our experimentation to come up with better solutions for applying automation to things like tissue culture. We had planned to talk exclusively around cell line development applications. I do speak to that later on in this talk, so if you stick with us, we'll talk about how we can apply automation to cell line development and the kind of impact that that actually has, as well as a couple of other applications as well. So moving into that main content, I should probably say that I'm not a cell biologist, unlike Jeff, who is the scientist within my team focused on that area. But I have developed a skill set around applying automation to various different scientific applications. And this is kind of leads us more into the content for today about how we can redesign experimentation to make it more amenable to automation, and particularly in the cell biology space. For the stage we're at in terms of standardization of experimentation around tissue culture-based experimentation, there really isn't as much as we think we should see in that space. And I've got a few questions about why that might be. First of all, I want to think about some general questions, and then I'm going to go into some specific things about why this might be particularly challenging for tissue culture itself. So one of the first challenges we see routinely is one of lab space. This is a headline from a Financial Times article from last summer, but still very representative of the challenge that we see of lab space shortage getting to such a state that it's actually threatening the boom of a particular industry in the one of life sciences and biotechnology, which is quite astonishing that we've gotten to that stage. And the reason why I raise this in the context of automation is that one of people's first reactions to automation is I need to make space for this to happen. Something has to change in the space that I have to be able to place automation within it. And I want to challenge that ever so slightly. First of all, that may be true, but it also may not be true. I think one of the things that automation can do, first of all, is really unlock the potential of a space that you already have, and you'll see some examples of this later on, and perhaps even maximize your output so you get more experimentation done per square meter than perhaps you would be able to achieve without automation in place. We also see these fairly consistent barriers to automation that are very generalized, that are really based on emotion and experience, and kind of the fear factor of driving this into laboratories. And I'm in particular thinking about laboratories that really haven't had a huge amount of automation experience. So a lot of people consider automation to immediately be these huge sort of monolithic systems like the one on the right here is a very generic automated system. It doesn't really matter what it is. just representative of something massive. And some automation might be huge. You might need to be significant in size and scale to achieve the goals that you want to set out to be, but it certainly doesn't need to be that way. And I certainly wouldn't recommend that scale of automation to be your entry point into this world either. I think it's far more amenable to people to start off with something a little bit smaller and move on. But at the other end of the spectrum, most people's starting place for automation in the laboratory space is around liquid handlers. Let's face it, most life science activities involve moving liquid from one place to another. And so we tend to steer towards liquid handling as our first port of call of automation. And there are plenty of people that have had an experience of not getting that quite right, or hearing about people that didn't get that quite right at the outset. There's probably more successes than failures in this space, and I speak as someone who comes from a very liquid handling centric automation background. But if we either go for something too complex in the top left, or something less complex than we actually need, then we end up having these systems sort of gather dust on the side, and they glean a very bad reputation as a result. So I think there's a lot of psychological challenges about adoption of automation that are very generalizable in this space. But something I want to think about in particular is, in the cell biology space, and more than anywhere else, we see this huge impact on the lack of automation on being able to take advantage of our experimental planning space. And what do I mean by that? And it's really a question of time. So I can't put a number to it exactly, but it's a very large proportion of the clients I've spoken to and consulted with about automating tissue culture in particular, or tissue culture centric experimentation. They always say the same kind of statements. I always start my experiment on insert day of the week, so that I can avoid coming in at the weekends as much as possible, which I totally get. I mean, who wants to be in a laboratory over the weekend, gazing out, especially this time of year when everyone else is having a joyous time in the sunshine, when you're sat there with your hands in a tissue culture hood, carrying out media exchange. I completely get that. And so unfortunately, we're faced without automation of having this compromise between a good work-life balance, or good experimental planning and good experimental and good usage of that experimental space that we have in terms of time. This is, and of course, in some countries, this is an enormous challenge, because actually your work contracts may prohibit you from working out of hours at all. And so we immediately are really not taking advantage of the real potential of our time, or our space, or laboratory time, if you want to think about it that way, without utilizing automation. So let's think a little bit more, we go a little bit further into the weeds, and really talk about some of the actual challenges about adopting automation for cell biology at a slightly more granular level. The first thing I want to speak about is labware. And I'm going to speak about the problems first of all, and then I'm going to come back to where I think there are some real tangible solutions to this. So first of all, and you'll see most of my conversation today is going to be about adherent cell culture. And that's where we see an abundance of these very recognizable TC flasks. And you can see, even just from this image, some of the challenges that presents. We as a human have to hold them in a very particular way, in order, especially if we want to get all the liquid out, using a pipette that's very long and has to go through quite a narrow neck. So there's all these challenges presented by that piece of labware, which, of course, they are solvable through automation. But I'm going to question whether that is the right way of solving these challenges as we go through this content, because building automation that is really specific to a piece of labware without considering about whether we can change something to take advantage of all of the off-the-shelf devices that are out there that deal with other kinds of labware seems a little bit short-sighted to me. Of course, these pieces of labware also utilize closures that are normally some kind of screw cap, and again, we can automate them ourselves at Automata. We built a screw decapper, but if we can avoid doing that, that takes away a process step that otherwise might not be required. Just a brief word on the pipetters here. I think finding an off-the-shelf pipetter that can handle that kind of volume or length of pipetter in an automated world is again not impossible, but it is very challenging and very limiting. A little bit about containment as well. I was going to speak about this picture earlier on, and I moved it to here because I think it's more pertinent, but this is the classical image that comes to mind when you think about a bench scientist doing some form of tissue culture, sat with their arms poking through a hood, hour after hour, moving liquid around using long pipettes. And when we think about that kind of containment, that is ubiquitous in tissue culture labs. But I would also ask us to question, when we think about automation, whether we need that level of containment, because immediately it constrains the kind of system we can build in some form. And so we always have to ask that question about what am I protecting here? And is my product actually safe? And so I just want to keep it sterile, or does my product, as in my and what I mean by product is my cells that I'm culturing or what they're producing, hazardous in some way, in which case I do need to also protect my people. And right now we have an awful lot of conversations where people default to the statement, well, it must be the same level of containment that I'm using in the lab, and then actually with further explanation, perhaps it doesn't need to be. Perhaps it doesn't need to be. So this ties very closely to the labware itself. And I'm going to steal a video from Gibco, now part of Thermo Fisher, where they speak about the basics of handling cells, and in particular about passaging cells. And you can see as we go through this, the kind of motions that are required. So these complex human motions that are hard to recreate in automation, well, not impossible, just difficult. So here we're tilting labware as well as going in with that long pipette, and we've got two hands moving things in a three-dimensional space. As we go into the next part of this process, you can see once the wash buffer has been added, the PBS here, that the cap is then returned, and then the labware is rocked and shaken in order to give a wash. So these are all things which we need to think about how we're going to recreate in an automated environment, and what's going to make those more viable to do that robustly and routinely. Next, I want to touch on media. So media is a really interesting one, and often overlooked when building out solutions for tissue culture. So if you think about what we do in a manual lab when it comes to media handling, and again, so many of these things like the motions we made, we kind of do subconsciously. We get so used to doing them, but we'll go to the fridge some period of time before we actually want to carry out the usage of that media. And we'll get the media out and we'll put it into our tissue culture hood. And the reason why we do that is so that it can warm up to room temperature so that it's ready to use and we don't shock our cells. So we have to think about that when we're building automated systems. There is a temperature requirement for storing that media, i.e. it's going to be cold, but then it's going to be warmed up just in time as we bring it to wherever we're going to utilize that, be it on the liquid handler or through some other device. And so we can't necessarily do it in the same way that we would do it manually, because we're not going to get out a bottle of media from a fridge and leave it on the side for a period of time and then put it away. We need something that can be used at a much larger scale than we would do manually. Speaking about scale, one of the main drivers for automation is throughput. If your goal is to up your throughput, then your media consumption is going to go up, most likely, which means you could end up with huge quantities and liters of media being utilized. And where do we store that? And of course, that means we have liters and liters of waste as well. And where does all of the waste go? And we have to consider what happens with that. And then there's this other layer of complexity is a little bit different, which starts making is more of a process problem when it comes to automation. But let's say we have basal media into which we want to add some kind of additives to make up specific variants of that media for different cell lines on the fly. That's something we might do quite naturally in a manual environment. We can certainly build into automated systems, but adds an awful lot of processing complexity where we need that volume of media to be delivered to a liquid handler and then have those additives ready and probably held at some temperature to be added into that media as well. None of these things, again, impossible. They're just things we have to think about. Excuse me. So the other area I want to touch on is analytical devices, and this is a particular favorite of mine because this all ties back to labware at some point. So we have to think about how we're gathering data about our tissue cultures, our cells today. And if we're doing, if we're in a lab which has no automation and no devices which are very automation friendly right now, this is probably quite a familiar scenario where we have a slide-based microscope that we've had to do some manual preparation for, and then we're gathering data through the human operator and making decisions based on that data. So this is something that clearly might need to change as we move into an automated environment. And I'll come back to this a little bit later on. Now, everything I've said to this point has been around adherent cultures to some degree. I want to speak about suspension cultures really briefly. It's not my main focus here because most of the projects we have are typically adherent cells. However, suspension cultures present some of the same challenges and then an entirely different set of problems, mainly around the labware in the form of shake flasks or shake tubes, or these kinds of Erlenmeyer flasks that we see here. Their environment tends to be different. So here we can see some fairly standard, I never get the right name for this, but that bed type shaker, which there are actually emerging automation friendly versions of, but to recent times haven't been automation friendly at all. And of course, present a bunch of process challenges. So separating my cells from my media in a suspension environment is actually far harder than with adherent cells because the adherent cells are already stuck down. So I need to think about all those extra process steps, such as centrifugation, that I need to incorporate into that. So I will come back to this a little bit, but it's not something I'm going to go in super deep on today. So how do we address this stuff? How do we reshape those experiments to allow us to build automation? If I'm right and all these things are challenging for us, then what can I do in terms of my experimental design to adapt them and make them more automation friendly? And really, the reason why we'd want to do that is to make it more likely that our adoption of automation is successful. And going back to some of these challenges, I think when we spend a lot of time building deeply customized systems and a lot of like bespoke widgets, that tends to drive the higher chance of failure, both a failure point within the system and a failure in terms of adoption, because the implementation time tends to go up. So what do we have to think about here? And I tend to bucket these into a couple of different areas when trying to think about how we might change our experimentation. One is like considering what are my real physical constraints, i.e. the space that I'm working with, the labware that I'm working with. And the other area that I'll go into more, I'm going to go into more detail on the next slide about what are my real scientific constraints. I've got a few examples of those. And really the point I want to make here when thinking about my real scientific constraints is that I know I was guilty of this, and I see this so often when we go into labs, is we do things in laboratories because it's the way we have been doing it. Typically, we have an SOP that's been optimized at some point in time and we follow it, or we more often than not take that SOP and we have our own slight tweaks on it. But we often do things just like, for example, operating in those class 2 hoods. We do them because it's the way it has been done. And I think we should always question that when thinking about a major transition like adoption of automation, about what are my real constraints here, and really when we extend that out, it's really like, what are my goals? What am I trying to achieve? So one of the first things I always think we have to get people to think about is my experiment currently constrained by manual operation? So is the goal that I want to achieve actually being inhibited by what I do manually? And one of the simplest examples of that relates back to the throughput challenge. Not all, but a lot of cell line development experiments are a numbers game. We might want to pump in a certain number of clones at one end to maximize the chances of getting really high performing clones out at the other end. And so this might create a scenario which just isn't possible manually. And we've probably been thinking about things the wrong way if we've been trying to achieve that manually and trying to hit that goal of upping throughput without thinking about how we might change things to drive automation. This one I really enjoy because I think it also comes back to this labware thing in a way, but it's that question about like how many samples. So this might challenge the concept of throughput. So how many samples do I need to put in to actually get out the answer I need? How much volume do I need, or how many cells do I actually need? I think we saw a really good example. In fact, I've seen this more than once, where we've gotten to the question about, okay, we're culturing cells in a certain size of flask. How many cells do we get from that? And the answer will be a couple of million cells. And then we'll get into the question about what do we need in terms of numbers of cells to actually achieve the outcome. And it might be in the order of a couple 100,000, which of course then immediately challenges whether we needed that volume of cells in the first place. So we save on media, we save on processing time, and we also perhaps enable us to use the different kinds of labware that I'm going to come on to in a moment. And how am I gathering data now, and what could this be? I think this is another really interesting one for me, where we are often constrained by the time we have when we're currently carrying out an experiment. And that might force us to only gather a certain data set. And I'll speak about cell line development a little bit later on, but I think when you speak to people doing that kind of experimentation, so seeking out a high-performing cell line from a pool, if you ask them what they could do if they waved, what would you do if you waved a magic wand? It would be, I would love to have a larger data set to make a decision on, but I just can't do that manually. And automation might enable you to be able to do that. So I think it's worth thinking about right now about what data am I gathering, and what would I like this to be in an ideal world, and come back to what are my goals here? What maybe my goals are to gather a deeper data set in a way that I couldn't do manually. So coming back to this, where do I start? I think labware migration solves so many things. Again, we can automate around tissue culture flasks, but they do tend to present one of the largest challenges to automation. The manipulation is challenging. The pipetting is challenging. Also the limitation to analysis equipment is huge. There are automated devices that will work with some types of flasks, often the smaller ones that are very close to SBS footprint labware, but anything else might require some kind of bespoke device to enable that, or limit your pool of choice to something very, very small. We tend to encourage people wherever possible to migrate to some kind of microplate footprint labware. And perhaps this isn't a bigger compromise as you might think. Two 1-well plates here that you can see an image of is almost equivalent to one T175 flask. So actually the compromise is not necessarily huge. And the pushback we often get at this stage is, yeah, but now I've got 2 bits of labware I have to process. But my question would be at that stage, do we need to care? If we're using automation to solve this problem, do we care whether it's 2 pieces of labware or 1, or 4 pieces of labware or 1? We might do, and again, it comes back to our goals, because it might be that we have a particular throughput constraint and we're making this worse by having more pieces of labware, but more often than not, that isn't the core problem here. And so actually, I think migration to labware, yes, it does require some revalidation and retesting for a lot of people, but it tends to be the better way forward, because it unlocks so many of the other things I'm about to talk about. Again, this shift to automated devices, I'm gonna steal a bit more of the Thermo Fisher video around cell culture basics, and this one is great because it shows the manual process for doing cell counting and cell viability checking, where we load up a slide and we stain it with trypan blue, and then we manually count with our little clicker our live and dead cells. Clearly, we can't do this at any kind of scale. And so we have to think about what kind of device. A lot of people are doing this, right? I'd be astonished if the people in the call aren't actually already using some kind of automated cell counter, like a Beckman cell is one of the most common ones out there. But that isn't the only example. I showed a manual microscope earlier on, which might be used for other measurements, such as looking at our confluency. But there are plenty of different kinds of automated imaging devices out there right now, depending on your needs. The one I'm showing here is the Synentech CellaVista. I tend to think of that as a relatively low magnification plate-based imager, where we can look at wells and look at confluency and start making decisions based on that data. But there are other devices that will work at a much higher magnification, or perhaps there are other kinds of analysis that need to be done, such as flow cytometry or something like that, which now is also there are now also plenty of automated devices out there that will work around microplate based labware, which is why that shift to different kinds of labware starts becoming increasingly important, because now my pool of analytical devices goes up extraordinarily. So come back to suspension cells really briefly. I do want to make the point that in many cases, at a certain scale, automation of suspension cell culture is absolutely possible within these kinds of systems. So just because we were using Erlenmeyer flask before doesn't mean that we can then think about shifting to something that is SBS footprint. There are greater challenges, but they're not insurmountable. Often it comes down to whether we can handle a volume that's required. And you can just see here, this is the Thermo cell selector incubator, which allows us to shake at a reasonably large throw for automation. So I think it's a 20mm orbital shake. Reasonably large volume piece of labware for this well. So this is a very deep 6-well plate that can hold several tens of mils of culture. And so all of a sudden, we're starting to unlock the possibilities of working with suspension cultures. I don't have a video footage of this, but I showed the Kuna incubators earlier on, or that type of incubator, and there are now automated versions of those as well, which have automatic doors on and lock the bed into place, so we can access them with a robotic gripper and start loading up other pieces of labware into this automated system. Of course, there are all these other process steps we have to think about as well. So I include a picture of a centrifuge here because we may well have to pellet, almost certainly we have to have a centrifuge on these systems to pellet cells and allow us to do things like media exchange and splitting and cell counting. But those devices are available, especially when we switch into an SBS footprint labware centric world. So containment, I've spoken a reasonable amount of this already, but I want to come back to this about this. What does the implication actually have once I've thought about what my needs are for containment? What does that actually mean to us from an automation design perspective? And I just wanted to show two examples here of where if we do need just product protection, we can then continue to design quite open and user-friendly systems that also help to drive that adoption, because now I've got an open system with lots of these devices accessible and walkable too, because all I need to do is blow HEPA filtered clean air over that system at a particular rate. If I need product and user protection, this is very doable. I say around about half of the enclosures we spec are of this type, but you can immediately see we're building a much larger box around the platform, and we have to compromise in some other areas, and perhaps we do start turning the system into a bit more of one of those large monolithic systems. And we have to think about how we're going to make sure we test for that and make sure we drive good adoption of it. But these things, again, they're all solutions that have to be specified as part of the solution development process. So taking all of those things into account, so a lot of the problems that I've talked about are solvable. There are still going to be things that I need to build to make my system functional. And there are two areas in particular I want to touch on that are the most common ones that are left over. One of them is that labware manipulation, so the rocking of labware or the tilting of labware to be able to get large volumes out. Now, this is something actually, there are some commercially available tools for doing this, and often they're part of the larger liquid handler that we might integrate into a more holistic system. So this is a reasonably solved problem, but it is something we have to consider, and certainly as a slightly more bespoke piece of equipment that needs to be incorporated. The other piece, which requires a lot more thought and a lot more specificity when it comes to a particular individual client system, is how we're going to handle the media. There are some things which just remain pretty much always true. We're going to have a fridge to hold our media cold, and we're going to have something that warms up that liquid as it enters our liquid handler or dispenser. But this is going to be something that, whilst we have a standard toolkit for, is going to be specified to the system based on the volumes of media, the numbers of media types, and the devices that is being integrated with here. And so you can just see on this schematic here for one particular system, we're delivering 2 media types to 2 different reservoirs on the deck of a liquid handler that are being warmed up in process. And then we also have a dispenser, which is having one of those medias pumped through it, but also has to have the option of having a wash buffer pumped through it to be able to clean it of media as well. So there's a bunch of different things to think about, but this is the kind of thing we would work with you as part of our solution development when specifying an automated system. But at least at this stage, we've now limited the number of choices we have to make about really specific customizations to just a handful. Because by changing a few other things in our experimentation, we're now able to take advantage of this huge wealth of plate-based devices that are out there for all of the other functions we need to process a tissue culture experiment. So moving into the last section of my content today, I have to talk a little bit about our platform and how we are approaching some of those system designs and some of those challenges for cell biology experimentation. So for those of you that are joining that aren't aware, I'm not going to go super deep onto our platform in this content, but just to share with you what it is that we build at Automata. So we provide what we regard as being the first fully automated lab bench. We were looking at the approaches to automation right now, and we're thinking, we're pretty sure we can build something which makes better use of space, is more human-robot interoperable and easier to adopt and more modular with it. And so we chose to build our platform around really taking our standard bit of lab furniture, the lab bench, and building automation around that. What that actually looks like is there are three main components in the Link bench. We have the bench surface, which is our main integration space, where we're going to place most of the devices that we integrate together to build all the capabilities for an end-to-end process. We utilize a SCARA robot, which is a 4-axis robot that will pick up labware and really tend to those devices. And this is our main area for this robot, is to tend to the devices. And then the third part, which is very unique to the Link system, is the transport layer. So this sits just below the bench height and is our main labware logistics component. And I've got a video I'll share with you in a second just to talk that through. But in essence, this transport layer is there to move labware around the system and have it just in time in front of the robot, ready for the robot to deliver that to device. And with that parallel transport, we can start achieving throughputs that you otherwise wouldn't think were achievable. Just to share with you a little bit of video footage of that transport layer in motion. Here we can see one of the pucks carrying a piece of labware over the surface of that transport layer, moving it into position just in time for that robot to come and pick it up and move it up to the surface of the integration space. In this case, is actually moving it onto the deck of a TCAN liquid handler. So most of the systems I'm going to show you today are not all of them are quite linear in their format. We actually unfold them in that way to make them easier to explain. But you don't just build linear systems with the Link bench. We have had several other configurations we work with. So we do have the linear configuration, and actually this image also shows you a version without the transport layer if we need a higher vertical density of devices and we don't necessarily need that extra logistics. We also then have two forms of corner benches. We have an inside and an outside corner that can be utilized to then go around the edge of a room and start building into the shape of your laboratory. And of course, finally, we have this back-to-back configuration where the transport layer is critical for passing labware from one side to the other of this platform, but allows us to build systems which actually, if we're building containment around them, these tend to be a little bit more straightforward to build that kind of class 2 enclosure for. But also is that more conventional island-based automation if you've got a large space and that can accommodate it. Our software platform is a cloud-based software platform for doing both workflow development, workflow simulation, and also monitoring runs that are executing upon your platform locally. We can get into a deeper conversation about the software if you'd like on a further occasion, but everything is very graphical and we're building out the usability and intuitiveness of this as we go forward in time. So a couple of specific use cases. I always like to start off this by talking about some of these core tissue culture operations. So if I think about automating any cell biology experiment where I am culturing mammalian cells, I always have to do two things. I have to exchange media or feed my cells, and I have to do passaging or splitting. So media exchange, quite simply, where I'm going to get cells from an incubator, remove old media, either all or some of it, and then replace it with new media before putting the cells back in the incubator. So a super simple process. And more often than not, I do layer in some kind of analysis into that process as well. The other core process is this one of splitting. There are a few variations on this we see with different clients, but typically we're removing media, we're adding some kind of reagent to unstick the cells or dissociate them. So most commonly trypsin, could be acutase, doing some kind of incubation and then neutralization process. Some clients want to count the cells at this stage and do the splitting based on a cell count, and some people just go straight on to the next step and transfer a subpopulation based on volume into the new labware, put them back in the incubator. So we see a bit of variation here, but the capability requirements are pretty much the same. And this is quite a compact system that we've designed for delivery on all of those capabilities. So it is fairly liquid handler centric. So we rely on a reasonably high-end liquid handler. In this case, you can see a TCAN Fluent. We might also work with a Hamilton Star or a Beckman Biomech for this particular application. We also need an incubator. So we work with both thermo and laconic in this space. And then you can see on this platform, we've got a couple of analytical devices as well. We've got the imager I shared earlier on for measuring confluence of those cells. And then we've also got a cell counter, which actually in this case is the PerkinElmer or Nexcelom Silica MX for doing reasonably high throughput cell counting. And this platform on its own starts to unlock that ability to be able to run things in a completely walk away fashion for those basic media exchange and cell maintenance operations. And we start seeing when we do some utilization analysis with clients that I've got fivefold here, but actually fivefold is sort of a minimum potential increase in utilization when we start thinking about running those systems when we leave for work at the end of the day or even into a weekend. The next example I want to talk about is cell line development. In this example, we're seeding cells using some kind of single cell dispenser. And then over a period of time, measuring the growth and productivity of those cells and making decisions on that data to select for the high performers. Those high performers, we then expand typically into a larger piece of lab, so we might go from 3 4-well plates through 96, 24, 48, 6-well plates, or whatever your choice might be. Increasingly characterizing those cells before getting to a stage of having a smaller number of hits and freezing them down. Coming back to something I said earlier on, one of the big questions we get asked at this stage is, oh, well, how can I expand that analytical data set? How can I make a better decision or even just gather a deeper data set so that I've got more information about those cells as we go through the process? If I'm currently just making a decision based on growth and productivity, perhaps I'm not making them the most informed decision about which cell line's the highest performer. So here we see a system that we've unfolded. So I have made this very linear. That has a lot of the capabilities built into it for a very end-to-end cell line development process. So we see those core capabilities of liquid handling and incubation and cell counting onto this platform. So now we have the Beckman Coulter Vicell integrated. But we've then started adding in some other capabilities. So we have a single cell printer to work up the very top end of this process and dispensing a single cell into every well of a plate. We then also have an imaging system that is now a little bit more tailored to cell line development, measuring clonality as well as some of those core measurements about confluence. And further to that, in this case, we've added on a device for carrying out some productivity assays or product characterization assays. In this case, a Sartorius octet. On this system, it was really just there to measure product titer, but we also looked at whether we might start adding in analyses around glycosylation or charge variance or things like that, all possible through this kind of device. Finally, on the right-hand side of the system, we also have a further small liquid handler, really just for handling the pooling of cultures of clones into a single cryovial so that we can then store them ready for handing over to the next part of the process. And something we saw here is obviously the main focus of the system, actually the main goal of this kind of platform is to really up the throughput. But the thing I wanted to highlight here is when we got into that, what would you do if you could wave a magic wand discussion, we started thinking about how we would add on additional analysis devices to get beyond making decisions just around growth and productivity and start thinking about, well, could I look deeper at the cell health or the morphology of those cells and the clonality and different data about the product quality as well, and make a much deeper data set around the clones that we're selecting. The final example I want to speak to you is one around assay ready plates. So in this case, this isn't about growing up cells to go into multi-well plates to be handed over to a screening team. So in this case, we have a portion of our platform dedicated to maintaining some cell lines, so 10 or 20 different lines, a couple of pieces of labware each for redundancy, but really just going through that standard feed and splits process. And then at some point, we are going to request, or a user is going to request for some assay plates in a few weeks' time. And that will trigger some further activity around growing up more cells. So in this case, we're going from 2 labware to 12 pieces of labware to get enough cells to be able to dispense into every well of 196 well plates. Once we have enough cells, we then have to dissociate those cells, pull them, count them, and then seed them at the right density into all of those plates. And this bit of the process is the tricky bit, because this is where we become time constrained. As soon as we dissociate those cells, their health may start deteriorating. We want to get them back into a nice environment, those 96-well plates, to start recovering. The system for doing this in this particular case, I've got an island system. So this is 4 benches on one side and another 4 on the other, back to back. And you can see a lot of duplication on this system, and I'll explain why in a moment. But in general capabilities, we have liquid handlers, we have a cell counter, we have a whole load of delidders because we're working with a lot of lidded labware in this particular case. We have ambient consumable storage, some small incubators just for handling some of those cell treatments for dissociation, and a centrifuge. We also have 2 large capacity Lyconic incubators here, one for handling our input cell lines and one for handling our output assay plates, so kind of an in and out incubator scenario. And between those, we house our media fridge. And what's really interesting about this system, so the general capabilities for maintenance were pretty straightforward, but hitting a really stringent timeline was what drove us to have to double up on liquid handling and delidding capability on this platform when we looked at all the bottlenecks of it. And in this case, we're able to go through that process of dissociation through to plating out all of those 100 assay plates in under 90 minutes. So if your goals were different, if you could do that in a longer period of time, or if you needed less plates, then perhaps you'd be looking at a system half the size of this one, or something in that order. So that's the last thing I want to share today. I'll just leave you with a video of our demo system that is running in our London office. You can see our arms moving simultaneously and the transport layer moving labware just in time. And just close with the thought that what I really wanted to speak to you today was this idea that automation for cell biology is absolutely possible, but we will make our lives a lot easier if we start going back to the first and also first principles of that experiment, exploring whether to hit the goals that we want to achieve, whether there are experimental changes we could make to facilitate that automation adoption. Thank you very much. And with that, I can take any questions.
Thanks, Russ. That was great. Yeah, we've got a few questions in the chat, so we'll just run through them now. The first question is, biology is somewhat variable and a fixed timetable might not work for my cells. Could I get an automated system to start a process based on real-time data, such as a cell number or confluency?
Yes. The short answer is yes. There is, as always with these things, there is complexity to it, and it depends on exactly what you want to do. But yeah, we have designed systems already which are taking data. In fact, we have a reference site in the Crick, who incidentally there's a webinar about that system next week, which is a completely different space. It's a genomic system, but it's running QC for a sequencing process, and so we're actually taking data from plate readers, adapting that into a new work list for the liquid handler, and then driving a new set of operations. And that's a fairly simple version of what we're describing here, which is data-driven automated steps. So we can do this in the tissue culture world as well, where we can take data such as, I think confluency was measured here, or mentioned here. We can take confluency data and then make decisions about what we want to do. And in fact, and that last system I shared is also an example of where we've standardized an experiment for growing up enough cells to hit that 100 assay plates volume, but actually the exact number of cells we get is a little bit variable, and so we do go through a pooling and counting process to make sure that we are going to dispense the right amount of cells into every single plate. So that's another example of adapting to the biology based on the data we're gathering.
Nice. We have another question, which is, what other kinds of analytical devices can be integrated to these kinds of systems? And could we incorporate our existing devices into an automated system like you described?
Yeah, that's a great question, actually. So I mentioned a few here. I mentioned cell counters and imaging systems. I mentioned the Octet, which is a sort of bioanalyzer. There are a whole bunch of other devices that could be incorporated. I think in the world that I tend to live in, we tend to focus on the ones that are able to take microplates. The simplest of those is the microplate reader, so a multi-mode reader measuring absorbance, fluorescence, luminescence. Interesting, a lot of those can actually incorporate some imaging capabilities these days. But the other kinds of devices we see very frequently in this space are high content screening platforms, so much higher magnification images. They tend to have a little bit of a time differential in terms of read compared to the other devices we're talking about. So they might be reading a plate for tens of minutes or hours even. that raises a different sort of challenge. And flow cytometry as well is another one that comes up more frequently, but now there are quite a lot of plate-based and higher throughput flow cytometers that are more amenable to integrating, but of course they then also require a bit of extra sample prep as well, so we have to think about that in the process.
Thank you, Russell, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Automata, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 20th, 2025. Labyrinth will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.